My name is Ziynet. I'm a PhD candidate at University of North Texas and also research assistant at Dr. Sardar Bozda's lab. Today, I would like to present our project Supreme, a cancer subtype prediction methodology integrating multiomics data using Graph Convolutional Neural Network. I will start with breast cancer. It was number 2, but now it is number 1 most commonly diagnosed cancer worldwide. And in that cancer, we have some biomarkers which are leading different subgroups. And with those subgroups, they are same cancer type, but different subgroups. And these are guiding to different treatment decisions. Inspired from that, researchers studied molecular subtypes, which are differentiating characteristics in cancer. And in 2009, Parker et al. Discovered that expression of 50 genes are very important to the cluster breast tumor samples. And those are called as PAM50 subtypes and considered as ground truth for many studies. And then with increasing technology, various biological data sets from cancer tissues have been generated. For example, TCGA, the Cancer Genome Atlas Project, generated data for thousands of patients with 33 different cancer types. And in TCGA, we have 7 different data types for breast cancer. So in each data type, we have some samples, and each sample we have a vector for genes or loci. And considering that each data type contains a unique aspect of underlying biology, it's important to integrate those. So it is called multiomics integration. There are some studies which are doing integrative cancer subtyping. For instance, iCluster Plus does some dimensionality reduction and then combine the features and do clustering on it. So SNF generates many patient similarity networks, fuse them with an annular step, and then do clustering. Or PINS Plus tries to find clusters which are robust to data perturbation. On the other side, DeepCC tries to utilize gene expression with pathway information with deep learning. And the networks are also highly utilized to store information. So graph representation learning methods emerged. Simply, there's a network, and we are trying to get low-dimensional representations, which are called embedding. And there's a nonlinear transformation function, takes each node and map them to those embeddings. And the intention is that nodes with similar local neighborhood will go to close points in that low-dimensional space. And the recent concept is graph neural network called GNN. And GNN doesn't utilize local neighborhood only, but also node features as well. The intention is GNN aggregate information from neighbors using neural networks. And Graph Convolutional Neural Networks, which I will call GCN, is one popular architecture. It simply averages neighbors, including itself, and then apply a neural network. And GNN-based methods have been utilized for cancer subtyping too. Excuse me. GCNC, one of them, and it leverages one single network and does the convolution on just gene expression data. MogoNet tries to integrate the predictions from different GCN runs. But to properly utilize more from available knowledge, it is important to properly integrate multiple networks and multiomics features at the same time, simultaneously. So to achieve that, I will introduce our project Supreme, a node classification framework, but we specifically apply it to cancer subtype prediction problem, and we utilize GCNs. And while doing that, we apply convolution on multiple patient similarity networks. And on those networks, we encoded those networks with multiomics features that we have. I will briefly explain what it does. First, we are collecting data from all available data types. Here we have 7 data types, and we extract features from each of them separately. So from each of them, we have some patient vectors, which are features for that data type specifically. And then we build patient similarity network from each of them separately again. And here, the blue edges are showing the, you are not seeing, sorry. Here, the blue edges are showing the highly similar patient pairs in that blue data type, and red edges are showing the in that data type, and all of those are same. And after that, we compute patient embeddings. While doing that, we are utilizing GCN for each network separately. So our assumption is that if the patients are having similar local neighborhood, they will be likely belong to the same subtype. And here the similarity is based on our specific data types features. And also we called all those multiomics features as raw feature. I will call this raw feature. And while utilizing GCN, we utilize all multiomics features as our raw feature. So those are our not features in our GCNs. And after that, we have 7 different patient embeddings. And to extensively evaluate the performance, we run each combination of embeddings, meaning that I have 7 different data patient embedding, and I have 2 to the power 7 minus 1, meaning, which is 127 different models. So for each of them, we generate a model, and we integrated raw features. Why we did it? Because we are doing convolution, and in case their signals were diluted during the convolution, we wanted to add those as raw feature, and also I will show without that step too. After that, for each combination, we are doing some predictions, and we are predicting cancer subtypes, and we are feeding them into a multi-layer perceptron. We applied Supreme to breast cancer dataset from TCJ, Metabaric separately, and also we combine them. We are doing cancer subtype prediction task, and the grant rate is 50 subtypes. And I will show just the results on TCGA. Here we show the performance of Supreme against other cancer subtype prediction tools and baseline methods. Here the evaluation metric is macro-F1 scores. We have multiple models, so we are showing the violin plot to visualize the performance across all those combinations. And as I mentioned, to see the impact of raw feature integration, we also skipped that step and we called it Supreme minus. And according to our results, we are significantly better than other tools. And even though we don't have a significant difference between Supreme and Supreme minus, in most cases, Supreme outperforms Supreme minus, showing the importance of raw feature integration. In addition to that, I will show one more result. We are here measuring the ability to differentiate the samples based on survival. So we obtained p-values from each method and showing these p-values are showing the significance level of survival difference between subtypes. Here again, we have 127 different models, so we have this much p-values too, and we saw that we are consistently having significant survival differences. And it is mostly better than the grant rate, which is the dashed line. So to sum up, we developed this node classification network, which could be applied to any problem. We use the cancer subtype prediction problem. I will just mention that, interestingly, we were better than the grant route. And this might suggest that our grant route is just utilizing gene expression. And this might suggest that utilizing the other data types, Supreme could demystify the undiscovered characteristics in the data and in those cancer subtypes. And in that way, we can improve the grant route labels. And with those more clear cancer subtypes, it will help us to pave the way for precision medicine. I will have more results, but I will not be able to. But if you can follow those links, they will be publicly available soon. And I would like to express my special thanks to Dr. Serdar Bozdar for his supervision, my lab friend for their support. I want to acknowledge our NIH grant and also ISMB organizer for this opportunity. Thank you all for your interest.